Hey, hello, ladies and gents. Hope you're all doing well. Today, we are going to be doing activities related to homeostasis and how to maintain that. So, we look at the concepts of negative and positive feedback. Now, first, let's review the notion of homeostasis. Stasis means standing or staying the same, or stasis means standing or staying, and homos means same. So, your body's trying to maintain constant levels or conditions, whether it's something in the blood, body temperature, your heart rate. Everything tries to maintain steady states at whatever levels are typical for the body. And we do that through some feedback mechanisms. First, we look at negative feedback. Remember, negative feedback may sound bad, but it's actually good because negative feedback loops work to keep the body at homeostasis. And that happens because whatever you produce will actually work to limit its own production. Sorry, trying to move the image around here. So here we look at blood sugar. Your blood sugar maintains a level of about 100 micrograms per deciliter, I believe, is what the units are. So you try to basically keep it at 100. Think of it that way. Now, if you eat a meal and your blood sugar goes up, then you've moved away from homeostasis, right? So what will happen is your pancreas will release a hormone called insulin, and then insulin causes your blood sugar to go back down to 100. So again, keep it at 100, have a donut at breakfast, your blood sugar goes up, your pancreas releases insulin, and then your body responds by bringing your blood sugar back to 100. Now, what if you've gone 5 or 6 hours and you haven't eaten? Your blood sugar will start to drop below your set point. So your blood sugar goes below 100. So then the pancreas releases a second hormone called glucagon, which will actually raise your blood sugar back up. Okay. So again, the idea of negative feedback is that your body responds to bring it back to homeostasis. Positive feedback, the way I remember positive feedback, I think positive pushes away from homeostasis. So whatever end product is created will actually enhance its own production. I think in the first lecture we talked about childbirth. Another example of positive feedback is lactation. So the suckling of an infant is a stimulus. That stimulus is detected by the mother's body and causes the release of a hormone called oxytocin. Oxytocin makes the muscles surrounding the milk ducts contract, which causes the release of more milk. Well, more milk being released causes additional suckling for the infant, which causes more oxytocin release, which causes more contraction in the milk ducts, which causes more milk release. So you get this positive feedback loop where the feeding of the infant causes the release of more and more milk until the stimulus is removed, until the baby gets full and stops feeding. So again, just think positive pushes away from homeostasis. Now, looking at the structure of feedback loops, the receptors are located all over the body and they detect what's going on. In this scenario that you see here, the eye is detecting the stimulus, which is the water. The central nervous system, which is our brain and spinal cord, then makes decisions. It either recognizes the stimulus or it doesn't, and then it decides to act. And then the instructions are sent to an effector. And an effector is either a muscle or a gland because they do things. Muscles contract and move the body around or move things through the body. Glands release chemical messages or hormones. Okay. So the receptor detects what's going on, in this case, the eye, which is sort of weird. Looks like a big old eyeball sitting on a table. But the receptor detects what's going on. Central nervous system decides, and then the effector acts. It does something. Now, two additional important components to this are the wires, the biological wires that carry that information. The sensory neurons that bring that information to the brain are called afferent pathways. And the way I remember this, I think it goes at the central nervous system. And then the wires that carry the instructions out to the muscles and glands, those are collectively referred to as the efferent pathways. Those are motor neurons that carry electrical signals to your, again, muscles and glands to do something. All right, so what I'd like to do is give you guys an opportunity to see how your body responds to a deviation from homeostasis. When the body exercises, the muscles are moving, right? And muscles require a lot of ATP, and part of the ATP production process produces CO2. So when your muscles are active, you make CO2, and then your cells dump that CO2 into the blood. When CO2 enters the blood, it forms carbonic acid, and your blood starts to acidify. A lot of people think that when they're running and exercising, they start panting or breathing heavily, it's because their body's saying they need oxygen. It's true, you need oxygen, but the actual stimulus that is acting on your body is the drop in pH of your blood. So the CO2 that's being dumped into your blood by your muscles uh, causes the blood to begin to acidify. You have pH receptors in your bloodstream that are like, hey, the blood's getting acidic. We got to get rid of this CO2. So your body responds to that. So that you want you to think about how you get back to homeostasis. So what you're going to do in this activity is go to the homeostatic control demonstration document that's in this Canvas assignment, and it sort of walks you through what to do. You need to get a baseline heart rate, and you can do that by finding your radial pulse or your carotid pulse. So the radial pulse you find by taking two fingers, placing it on the inside of the wrist, and then sliding it over to the thumb side. So it's sort of hard to get situated here, but two fingers, wrist, slide it over to the thumb side and press. Or you can get your carotid pulse, where again, two fingers go to your windpipe and then slide it off to the side. So either way, whether it's your radial pulse or your carotid pulse, find your heartbeat, and then you'll record that for 15 seconds, how many beats in 15 seconds, and then multiply that number by 4 to get beats per minute. Or you can do it for 30 seconds and multiply it by 2 to get beats per minute. You could do it for 10 seconds, multiply by 6, whatever. Just get your heart rate in beats per minute, and that serves as your baseline. Then what you're going to do is some sort of physical activity. So a moderate physical activity. You can go for a brisk walk, climb upstairs where you may live, whatever you want to do that for 2 minutes. And then immediately following the completion of that 2-minute activity, take a second measurement of your heart rate. So we'll look at what your body does when you start to perform activity and move it away from homeostasis. If you have injuries or anything like that, please don't do anything. Don't participate. Everything we do in here is by invitation. You're invited to do things, but it's not compulsory. You're not required to do things. So we invite you to go and do the activity, get the experience, get the feedback, and then in that homeostatic control demonstration doc, there are some questions on the back page. I'd like you to just review that or think through that, and that can serve as a conversation or starter for when we are back in class. Now, your points for today are going to be in the feedback loop practice. That's a Google Forms document. It's also hyperlinked in the Canvas assignment. You'll go through that, make sure you select the appropriate teacher and class period, and then I think there are maybe like 8 questions that you'll complete. So you complete that, make sure your name's in it in the document, your last name, comma, first name, submit that, and then that'll be your points for today with everything. If you have any questions, just send me an email and we'll take things from there. Thank you very much, ladies and gents. Have a good day. I always struggle to try and figure out how to turn this thing off.